Hello and welcome, dear students. Today we are going to discuss about the topic population structure and admixture in human populations. The main objectives of today's lecture are to know about the basics of population structure, to discuss about the measures to understand population structure, to understand admixture in human population, to know about estimating genetic diversity and ancestry in admixed populations, to understand role of population structure in genetic epidemiology. Dear students, let's start with the introduction first. Population structure is defined by the organization of genetic variations and is driven by the combined effects of evolutionary processes that include recombination, mutation, genetic drift, demographic history, and natural selection. Population structure, which is also known as genetic structure and population stratification, is the presence of a systematic difference in allele frequencies between subpopulations. In a randomly mating or panmictic population, allele frequencies are expected to be roughly similar between groups. However, mating tends to be non-random to some degree, causing structure to arise. For example, a barrier like a river can separate two groups of same species and make it difficult for potential mates to cross if a mutation occurs. Over many generations, it can spread and become common in one subpopulation while being completely absent in the other population. Genetic variants do not necessarily cause observable change in organisms, but can be correlated by coincidence because of population structure. A variant that is common in a population that has a high rate of disease may erroneously be thought to cause the disease. For this reason, population structure is a common confounding variable in medical genetic studies, and accounting for and controlling its effect is important in genome-wide association studies. By tracing the origins of structure, it is also possible to study the genetic ancestry of groups and individuals. The basic cause of population structure in sexually reproducing species is non-random mating between groups. If all individuals within a population mate randomly, then the allele frequencies should be similar between groups. Population structure commonly arises from physical separation by distance or barriers like mountains and rivers, followed by genetic drift. Other causes include gene flow from migrations, population bottlenecks and expansions, founder effects, evolutionary pressure, random chances, and in human cultural factors as well. Even in lieu of these factors, individuals tend to stay close to where they were born, which means that alleles will not be distributed at random with respect to the full range of the species. Dear students, let's now discuss about the measures to understand population structure. Population structure is a complex phenomena and no single measure captures it entirely. Understanding a population structure requires a combination of methods and measures. Many statistical methods rely on simple population models in order to infer historical demographic change, such as the presence of population bottlenecks, admixture events, or population divergence times. Often these methods rely on the assumption of panmictia or homogeneity in an ancestral population. Misspecification of such models, for instance by not taking into account the existence of structure in an ancestral population, can give rise to heavily biased parameter estimates. Simulation studies show that historical population structure can even have genetic effects that can easily be misinterpreted as historical change in population size or the existence of admixture events, even when no such events occurred actually. Dear students, let's now discuss the heterozygosity. One of the results of population structure is reduction in heterozygosity. When populations split, alleles have a higher chance of reaching fixation within subpopulations, especially if the subpopulations are small or have been isolated for long periods. This reduction in heterozygosity can be thought of as an extension of inbreeding, with individuals in subpopulations being more likely to share a recent common ancestor. The scale is important as an individual with both parents born in the United Kingdom is not inbred relative to that country's population, but is more inbred than two humans selected from the entire world. This motivates the derivation of Wright's F statistics, also called fixation indices, which measures inbreeding through observed versus expected heterozygosity. Dear students, let's now discuss in detail about the admixture. Admixed populations are formed as moderate to large scale movements of individuals, which allows the exchange of genes between two or more previously isolated populations, creating populations with ancestors from multiple sources. Genetic admixture occurs when previously diverged or isolated genetic lineages mix together. Admixture occurs when individuals from two or more previously isolated populations interbreed. So the previously isolated populations are referred to as ancestral or parental, and the newly formed population is referred to as admixed. In non-human species, the same process is often referred to as hybridization. Admixture results in the introduction of new genetic lineages into a population. Climatic cycles facilitate genetic admixture in cold periods and genetic diversification in warm periods. Natural flooding can cause genetic admixture within population of migrating fish species. Genetic admixture may have an important role for the success of populations that colonize a new area and interbreed with individuals of the native population. Dear students, let's now discuss about the admixture in human population. One evolutionary process that requires special attention in both population genetics and statistical genetics is admixture. Gene flow between two or more previously separated source populations to form a new admixed population. The admixture process introduces ancestry-based structure into patterns of genetic variation within and between populations, which in turn influences the inference of demographic histories, identification of genetic targets of selection, and prediction of complex traits or characters. Ancestry in admixed populations varies at multiple genetic scales, with variance among individuals and within individual genomes. An individual's genotype can be modeled as an admixture between k discrete clusters of populations. Each cluster is defined by the frequencies of its genotypes, and the contribution of a cluster to an individual's genotype is measured via an estimator. In 2000, Jonathan K. Pritchard introduced the structure algorithm to estimate these proportions via Markov chain Monte Carlo modeling allele frequency at each locus with a disject distribution. Since then, algorithms such as admixture have been developed using other estimation techniques. Estimated proportions can be visualized using bar plots. Each bar represents an individual and is subdivided to represent the proportion of an individual's genetic ancestry from one of the K populations. Varying K can illustrate different scales of population structure. Using a small k for the entire human population will subdivide people roughly by continent, while using large K will partition populations into finer subgroups. Though clustering methods are popular, they are open to misinterpretation. For non-simulated data, there is never a true value of K, but rather an approximation considered useful for a given question. They are sensitive to sampling strategies, sample size, and close relatives in data sets. There may be no discrete population at all, and there may be hierarchical structure where subpopulations are nested. Clusters may be admixed themselves and may not have a useful interpretation as source populations. Genomic data have revealed the extent to which genetic admixture has been ubiquitous throughout human history and can shape the distribution of human genetic diversity in ways different from those predicted by classical population genetic models. Admixture is a process whereby gene flow occurs between previously divergent source populations, producing new populations with ancestry from multiple source populations. Variation in admixed populations can be summarized based on ancestry from source populations. These ancestry patterns may vary between admixed populations formed by the same source populations, between individuals within an admixed population, and across loci within an admixed individual. Population geneticists have long recognized that studying admixed human groups provides opportunities to learn about evolutionary forces. Additionally, in practice, defining admixture in humans is highly context dependent, affected by social structure that influence population or self-identification, as well as methodological limits on detecting admixture from genomic data. Discussions of genetic admixture are implicitly or explicitly predicated on the idea that meaningful genetic differences exist between discretized human groups. In practice, the term admixed can vary to encompass a range of spatial and temporal processes of gene flow between previously isolated groups. However, extensive gene flow between groups is a hallmark of recent human evolution. Nearly all populations can be described as descended from a combination of multiple ancestries. Methodological issues and patterns of genetic diversity are not the only factors shaping the understanding of genetic admixture. A long record of societies and scientists' use of large superficial characteristics to classify human groups also plays an important role. Geneticists and anthropologists have long wrestled with the various field-specific and lay definitions of population, ancestry, ethnicity, and race, which interact and intersect with each other in complicated ways. In discussions of human genetic admixture, it becomes especially important to emphasize that these categories do not map onto each other one-to-one, and that race and ethnicity in particular describe classifications based on social phenomena. Dear students, let's talk about estimating genetic diversity and ancestry in admixed populations. Before polymorphism data could be generated at a genome-wide scale, several methods of measuring genetic diversity had already been proposed, including heterozygosity and nucleotide diversity. By connecting these to theoretical population genetics models, summaries of genetic variation can provide insight into the evolutionary force acting on populations. The quality of ancestry estimates depends on a variety of sampling and evolutionary scenarios. A recent study of the admixed Ashkenazi Jewish population noted that the lack of differentiation between European and Middle Eastern haplotypes made accurate local ancestry inference challenging, reducing their power to infer the parameters of the admixture process and demonstrating the complexity in defining admixed populations, as these populations are often not considered admixed. Dear students, let's now discuss about the admixture mapping. Admixture mapping is a method of gene mapping that uses a population of mixed ancestries, that is an admixed population, to find the genetic loci or position that contribute to differences in diseases or other phenotypes found between the different ancestral populations. The method is best applied to populations with recent admixture from two populations that were previously genetically isolated. The method attempts to correlate the degree of ancestry near a genetic locus or position with the phenotype or disease of interest. Genetic markers that differ in frequency between the ancestral populations are needed across the genome. Admixture mapping is based on the assumption that differences in disease rates or phenotypes are due in part to differences in the frequencies of disease-causing or phenotype-causing genetic variants between populations. In an admixed population, these causal variants occur more frequently on chromosomal segments inherited from one or the other ancestral population. The first admixture scans were published in 2005, and since then genetic contributors to a variety of disease and trait differences have been mapped. By 2010, high-density mapping panels had been constructed for African Americans, Latino, Hispanics, and Uyghurs, that is ethnic group in China. Dear students, let's now discuss about the dimensionality reduction in population structure. Genetic data are highly dimensional, and dimensionality reduction techniques can capture population structure. Principal component analysis, or PCA, was first applied in population genetics in 1978 by Cavalier Sforza and colleagues, and researched with high throughput sequencing. Initially, principal component analysis, or PCA, was used on allele frequencies at known genetic markers for populations, though later it was found that by coding single nucleotide polymorphisms, or SNPs, as integers, for example as the number of non-reference alleles, and normalizing the values, PCA could be applied at the level of individuals. PCA transforms data to maximize variance. Given enough data, when each individual is visualized as point on a plot, discrete clusters can form. Individuals with admixed ancestries will tend to fall between clusters, and when there is homogeneous isolation by distance in the data, the top PC vectors will reflect geographic variation. The eigenvectors generated by principal component analysis can be explicitly written in terms of the mean coalescence times for pairs of individuals, making principal component analysis useful for inference about the population histories of groups in a given sample. Principal component analysis, or PCA, cannot, however, distinguish between different processes that lead to the same mean coalescence times. Multidimensional scaling and discriminant analysis have been used to study differentiation, population assignment, and to analyze genetic distances. Neighborhood graph approaches like t-distribution, stochastic neighbor embedding, and uniform manifold approximation and projection, that is UMAP, can visualize continental and subcontinental structure in human data. With larger data sets, UMAP better captures multiple scales of population structure. Fine scale patterns can be hidden or split with each method, and these are of interest when the range of population is diverse, when there are admixed populations, or when examining relationships between genotypes, phenotypes, or geography. Variational autoencoders can generate artificial genotypes with structure representative of the input data, though they do not recreate linkage disequilibrium patterns. Dear students, let's now understand about the demographic inference. Population structure is an important aspect of evolutionary and population genetics. Events like migration and interactions between groups leave a genetic imprint on populations. Admixed populations will have haplotype chunks from their ancestral groups, which gradually shrink over time because of recombination. By exploiting this fact and matching shared haplotype chunks from individuals within a genetic data set, researchers may trace and date the origins of population admixture and reconstruct historical events such as the rise and fall of empires, slave trades, colonialism, and population expansions. Dear students, let's now discuss about the role of population structure in genetic epidemiology. Population structure can be a problem for association studies such as case control studies, where the association between the trait of interest and locus could be incorrect. As an example, in a study population of Europeans and East Asians, an association study of chopsticks usage may discover a gene in the Asian individuals that leads to chopstick use. However, this is a spurious relationship, as the genetic variant is simply more common in Asians than in the Europeans. Also, actual genetic findings may be overlooked if the locus is less prevalent in the population where the case subjects are chosen. For this reason, it was common in the 1990s to use family-based data where the effect of population structure can easily be controlled for using methods such as the transmission disequilibrium test, that is TDT. Phenotypes, that is measurable traits such as height or risk for heart diseases, are the product of some combination of genes and environment. These traits can be predicted using polygenic scores, which seeks to isolate and estimate the contribution of genetics to a trait or character by summing the effects of many individual genetic variants. To construct a score, researchers first enroll participants in an association study to estimate the contribution of each genetic variant. Then they can use the estimated contributions of each genetic variant to calculate a score for the trait for an individual who was not in the original association study. If structure in the study population is correlated with environmental variation, then the polygenic score is no longer measuring the genetic component alone. Several methods can at least partially control for this confounding effect. The genomic control method was introduced in 1999 and is a relatively non-parametric method for controlling the inflation of test statistics. Genomic control is a statistical method that is used to control for the confounding effects of population stratification in genetic association studies. The method was originally outlined by Bernie Devlin and Catherine Roeder in 1999. It involves using a set of anonymous genetic markers to estimate the effect of population structure on the distribution of the chi-square statistics. The distribution of the chi-square statistics for a given allele that is suspected to be associated with a given trait or character can then be compared to the distribution of the same statistics for an allele that is expected not to be related to the trait. The method is supposed to involve the use of markers that are not linked to the marker being tested for a possible association. It is also possible to use unlinked genetic markers to estimate each individual's ancestry proportions from some k subpopulations which are assumed to be unstructured. More recent approaches make use of principal component analysis as demonstrated by Alkes, Price and colleagues, or by deriving a genetic relationship matrix, also called a kinship matrix, and including it in a linear mixed model, that is LMM. Principal component analysis, or PCA, is a popular technique for analyzing large data sets containing a high number of dimensions or features per observation, increasing the interpretability of data while preserving the maximum amount of information and enabling the visualization of multidimensional data. Formally, PCA is a statistical technique for reducing the dimensionality of a data set. This is accomplished by linearly transforming the data into a new coordinate system where most of the variation in the data can be described with fewer dimensions than the initial data. Many studies use the first two principal components in order to plot the data in 2 dimensions and to visually identify clusters of closely related data points. So principal component analysis has applications in many fields such as population genetics, microbiome studies, atmospheric science, etc. Principal component analysis and linear mixed models have become the most common methods to control for confounding from population structure. Though they are likely sufficient for avoiding false positives in association studies, they are still vulnerable to overestimating effect sizes of marginally associated variants and can substantially bias estimates of polygenic scores and trait heritability. If environmental effects are related to a variant that exists in only one specific region, for example, a pollutant is found in only one city, it may not be possible to correct for this population structure effect at all. For many traits, the role of structure is complex and not fully understood, and incorporating it into genetic studies remains a challenge and is an active area of research. Dear students, let's summarize the lecture now. Admixture between populations is a fundamental process that shapes genetic variation and disease risk. For example, African Americans and Latinos derive their genomes from mixtures of individuals who trace their ancestry to divergent populations. A study of the ancestral origin of the admixed individuals provides an opportunity to infer the history of the ancestral groups, some of whom may no longer be extant. The admixture history of a population, such as the timing and source contribution levels, leaves predictable patterns of genetic variation within and between individuals from the admixed population. Empirical genetic analysis can therefore be used to infer the histories that produced observed genetic variation. If populations are subdivided, they can evolve apart somewhat independently. Population structure allows populations to diversify. This is the reason why population structure is a very important part of evolutionary genetics. Dear students, it was all about today's lecture regarding population structure and admixture in human populations. Hope you have understood it well. See you next time with a new topic. Till then, take care. Goodbye.